Hi, my name is Harshita Keerthipati, and I'm a rising junior at Amador Valley High School. I'll be presenting a comparative genomic analysis of colorectal cancer microbiome bacteria on behalf of my lab. Our research revolves around colorectal cancer, which is cancer located in the colon affecting the large and small intestines in the digestive system by creating uncontrolled tumor growth. Many factors, such as daily habits, genetics, and the environment, may impact the development of colorectal cancer, but what is often ignored is the gut microbiota that plays a role in the onset of it. Our research focuses on the microbiome in the gut, specifically targeting bacteria. The gut microbiome consists of microorganisms, mainly bacteria, that live in the intestinal tract. Our research purpose is to target pathways impacted by shared proteins of both probiotic and antibiotic bacteria in order to decrease the metastasis and malignity of gut tumors by increasing the expression of inhibitors of colorectal cancer. Our research is also dedicated to helping the wet lab in their experiments to narrow down bacteria regarding the expression of colorectal cancer to help accelerate their experimental processes. We hope that by narrowing down bacteria associated with non-cancerous status in the gut, we can help find more efficient treatments for colorectal cancer. Our procedure has consisted of 6 steps, starting first with the basic research of probiotic and antibiotic bacteria through reading literature and researching NCBI's database. We found many genes of interest and isolated them to further look into their differences, as many of the bacteria had a cancerous status, whereas some had a non-cancerous status. From here, we downloaded each set of genes into RStudio to compare the bacteria to one another in order to find similarities and differences. We started with inner joins, which were used to find similarities, and then moved to outer joins, which was a method used to find differences among our bacteria. From here, we ran alignments of our probiotic genomes and our target proteins to find similarities. This was all executed through NCBI BLAST, where we then downloaded the areas of similarity to do further research. We currently have created 4 models regarding the phenotypic and genotypic outlooks of the bacteria using data availability. We would ultimately want to create more comparative genome figures using the bacteria. Moving on to the tools we used, we initially started out with RStudio, which is a computational programming app using the language R. RStudio was utilized in our research to download the genomes of our bacterial interests. The usage of RStudio allowed us to first narrow our target proteins down into a few that were common among our known probiotic bacteria. We then switched to NCBI BLAST in order to align the genomes of the probiotic bacteria to help us find structural similarities and differences that may play a role in how effective each probiotic bacteria is in inhibiting colorectal cancer. We are currently using an information system to compare biomedical data called BVBRC, which will assist in the creation of our figures. We have been heavily utilizing this public tool as it contains a multitude of tutorials and tools regarding genomic alignments and protein structures. Our research project was initially centered around the recently published research paper targeting the gut and tumor microbiota in cancer, and it contains a list of both probiotic and cancerous bacteria. Our main focus throughout this research was on probiotic bacteria and comparing one to another in order to determine similarities. We initially chose 3 main bacteria to focus on, but only ran alignments on 2 of them. We initially started with a small set of bacteria to test our methods on, and then we later implemented it on a bigger group of data. B. longum was a probiotic bacteria we researched, which is a lactic acid producing bacteria in the intestine that regulates intermediate biomarkers of colorectal cancer. Our next 2 probiotic bacteria were the main focus of our research due to there already being a lot of pre-existing research on B. longum. We decided to focus on T. sanguinis, which is a gut microbe that is found in the gut lumen expressing membrane proteins. We aligned T. sanguinis to P. copri, and high levels of P. copri are correlated with lower levels of microbiota that are previously associated with immune regulating properties. We chose to align P. copri and T. sanguinis due to their active role in the gut biome and their associations with regulating the body. When we aligned T. sanguinis to P. copri, we found a multitude of structural similarities between their genomes, shown in the table, showing that as probiotic bacteria, they had a lot of similarities. Moving on to our bigger set of data, we created 2 models for both non-cancerous and cancerous bacteria. Our first model is a phylogenetic tree. The phylogenetic tree shows common ancestry amongst the bacteria from which we hope to identify target areas and pathways between the bacteria. This model helps us study specific pathways and the number of base differences between the various bacteria. Our second model for both non-cancerous and cancerous bacteria is a pie chart. The pie chart gives us information regarding the bacteria and how many and which ones play a role in what type of process. It is important for us to analyze the pie chart because we are able to compare it to our non-cancerous pie chart in order to determine the similarities and differences between the two. This would also help us answer some questions, like what is the difference between cancerous and non-cancerous bacteria? One current piece of literature we have been referencing includes a comparative whole genome approach identifies bacterial traits for marine microbial interactions. Throughout the paper, we can see many different models comparing marine microbiota, which we hope to recreate through our research on probiotic bacteria in colon cancer. Some future steps we hope to take is to create more models with both genotypic and phenotypic data using probiotic bacteria to find specific genes of interest. We are also currently working on creating an MSA model, which is a multiple sequence alignment model, which will help us to allow and align our cancerous and non-cancerous bacteria to help us further determine the answers to our research questions. We also hope to gather more research and information that can ultimately help the wet lab create a probiotic drink against colorectal cancer. We would like to thank the Aspiring Scholars Directed Research Program for providing us with the opportunity to conduct this research and our advisor, Clinton Kuna, for his guidance throughout our project. Thank you so much.